So class, today we are going to look at blood grouping. We are going to look at what? Blood grouping. We are going to look at blood grouping, and there are always two ways how we can carry out blood grouping. The first one is we have ABO, blood grouping. Then also we have the rhesus blood grouping. But first, we are going first to focus on blood grouping, the ABO blood grouping. So let us start with ABO blood grouping system. So under ABO blood grouping system, there are so many ways of grouping blood, but the most important ones, they are ABO blood grouping system and the rhesus blood grouping system. Why are they so much important than the rest is that the most important reason as to why we focus most on these two blood grouping types is that they are more antigenic. They are more antigenic than the rest of the blood groups. So during blood transfusion like that, they can cause what we call blood transfusion reactions. So that's why they are more important. Being more antigenic makes them be the more important blood grouping systems as medicine or medical is concerned. So now let us look at the types of blood of the ABO blood grouping. We have the different types of blood grouping. We have blood group A, blood group A, we have blood group B, we have blood group AB, we have also blood group O. So if you are not blood group A, you are either B or AB, or you can be blood group O. Now, we are learning about these blood groups. Let us look at the different genetic composition is in these blood groups. Because if you are blood group A, it means that you are genetically you are either having antigen AA or you having antigen AO. If you are blood group B, it means that you either have B or BO in your blood group system. Let me use this very part. So if you are blood group A, genetically, because we want to see how these blood groups are formed. But before we go to that part, let us see the compositions of the genetic composition of these different blood groups. With blood group A, you can either be having A as your genetic composition, or you might be having AO as your genetic composition. You know, these are autosomes. These autosomes, these are genes we get from one partner, one parent, and another parent. One parent brings this one parent brings this one. So in this case, one parent brought this one, another one brought this one. But you can be you are called A just because this one is recessive, this one is dominant. So with those with blood group B, you can either have B, which are called dominant, or you can have B, O, whereby one is dominant, one is recessive. So with AB, you can have only A and B as your genetic composition. Then with O, you will not have anything. You will have 0 antigens. You will not, with the blood group O, you don't have any antigens on the surface of your red blood cells. We are talking of the surface of the red blood cells just because, let us see, in the formation you'll come to realize that, take the example, if this is the red blood cell in saturation, this is the red blood cell. Now the antigen we are talking of, this one AO, these ones, they appear on the surface of our red blood cells. How do they appear? Now, you know very well that we eat starch, we get carbohydrates from there. Now the carbohydrates are broken down into carbohydrates are broken down into because they are polysaccharides. Then from polysaccharide they continue up to the end product, which is the monosaccharide. They are broken down into the monosaccharides. So there is a deposition on the surface of the red blood cell. There is a deposition of this monosaccharide, but it is not only one. So there is a chain. It joins together to make a chain of these monosaccharides. So a chain of these monosaccharides, actually they are like that all over they are like millions all over the red blood cells. These ones have just done it quickly like this, but let us use this one. Let us use this one for the study. Now, this chain which has been made on the surface of the red blood cell, this chain, it is a chain of monosaccharides. And a chain of monosaccharide is called, the whole of this is called oligosaccharide. This chain of these monosaccharides is called oligosaccharides. So with these oligosaccharides, remember, this is not a mature. We are in the formation of now. We are forming a red blood cell, and we are in the formation of also the blood group of this person. The red blood cell will now come out together with the blood group of the person. How will it come out with the blood group of this person? First of all, we have what we should know, that every human being on planet Earth must have an antigen we call the H antigen. The H antigen. You know the H antigen. H antigen. Sometimes they call it H substance. So it is, you can call it either H antigen or H substance. So now, here you must have the H substance before you have a blood group. Now, This is an immature red blood cell. We are still in the formation. So being immature red blood cell, we have also the nucleus inside here. Now, this is our nucleus inside our red blood cell. An immature red blood cell. Of course, the immature red blood cell is the one with the nucleus. Now, in this nucleus, we have what modifies. There is a chromosome. Now, a chromosome can be, so it can be named like 16, 17, from 1, 2, 3, 4, 6, 16, 17, 18, 19, 20. So this is chromosome number 16, chromosome number 7, chromosome number 18, chromosome number 19, chromosome number 20. Yeah? So do we have chromosome, because it started with the chromosome. Okay, let's continue. Yes, so we can have chromosome 10, number 10, chromosome 9, 8, 7, 6, 5, 4, 3, 1, 2. 3, 2, 1, yeah, like that. So from 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So in this red blood cell, what modifies this one? We have an oligosaccharide. This is a carbohydrate, oligosaccharide. So it is being modified by this chromosome 19. Chromosome 19. The chromosome 19. It is the one which helps in the modification. On this locus, there is a locus on this chromosome 19 here, which contains a gene, and that gene is the one which ignites the formation of the fucose transferase. Fucose transferase. Now, this fucose transferase, once you see the word transferase, it means that it is an enzyme that works in transport. Transferase. So this works in transport system. So this enzyme, fucose transferase, transports what is known as fucose. Transports this fucose and comes and joins it to this oligosaccharide. Now, a combination of fucose and oligosaccharide. This is the fucose. When they tell you name this, this is the fucose. This is oligosaccharide, a combination of monosaccharide. So the combination of fucose and the oligosaccharide is the one we call the H antigen. You get me? You get me now? Any question? Okay, we can continue. Now, a combination of the fucose, which has been transferred to the oligosaccharide, the combination now of fucose and the oligosaccharide is the one we call the H antigen. You must know that every human being on planet Earth has this oligosaccharide. Every human being has this, and this activity takes place in every human being. Now, that is formation of H. This is formation. Yeah, formation of H antigen. That is the formation of H antigen. Now, if we have this as our H antigen, how do we say that somebody or so and so is blood group A? What makes this person to be blood group A, O, B, O, O, A, B? It's what we are going to look at. Now, formation of blood group O. Let us begin with the A. Formation of blood group A. Now, formation of blood group A, you know very well we have our red blood cell. We have our red blood cell, this one. Then, should we use this very one? Okay, let us make another one. We still have space. So, this red blood cell, it has its nucleus inside, because it is still in the formation. Now, it has a an oligosaccharide. So it has oligosaccharide all over. It has all over its surface. It has oligosaccharide. Now it has now got the what? There has been deposit transferring the focus transferase has already transferred the focus to join it together with the what? The oligosaccharide. Now this is our H antigen. But we want to form blood group A. This person is making blood group A. How does it happen now? How does it become blood group A? Now, here, there is an enzyme called, there is a chromosome now. Chromosome number 9. This chromosome 9. You know you inherit from the father a chromosome. Yeah, this is from either the father or the mother. This is also from another pattern. Now, these ones, they have a gene on this locus. This chromosome 19 has a locus which has a gene. That gene will ignite the formation of the what we call the N-acetyl galactosamine. N-acetyl galactosamine transferase. So this one ignites the formation of the enzyme called N-acetyl galactosamine transferase. This N-acetyl galactosamine transferase will now transfer the N-acetyl galactosamine and deposit it on top of this H antigen. Now, on top of this H antigen, now we have another deposition of another because this is also another sugar. So there is another sugar again being added, because we have two now sugars. We have the first one was the oligosaccharide, this chain. Now they added the 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 focus. It is also a simple sugar which has been added. Now the combination became H antigen. Now when this chromosome number 9 ignites the formation of the N-acetylgalactosamine transferase, and they transfer the N-acetylgalactosamine to the on top of this. Once the N-acetylgalactosamine has been deposited on top of this H antigen, it means that this combination has now become antigen A. And once it becomes antigen A, it is now called, it is no longer called, it is not just an H antigen, but it is blood group A. So this is how blood group A is formed. You get me? This is how blood group A is formed. So you inherit these ones. Even this part also is also doing its work here. It is also doing its work here. So this locus here has a gene. This locus also has a gene. This one from another pattern, this one from another pattern, from your parents. So it does its work like that. This one does its work, this one also does its work to produce antigen A. Now a combination of this H and A is blood group A. Now we want to see how blood group B is being formed. Now blood group B, you know very well we have our red blood cell as usual. It has this H, it has this chain, these chains all over it, this chain of the oligosaccharide. The oligosaccharide is, you know it very well, it is there. Now it has also, the oligosaccharide has the focus, so it is now our H antigen there. Now it is going to be blood group B. How? This same chromosome 9 being inherited from the parents, it will come, it has a position, a locus. This locus, these are now alleles, this one and this one, these genes. So these alleles. So the gene which is here will ignite, in this case it will ignite the galactose transferase enzyme. Galactose transferase. Now the galactose transferase will now transfer the galactose to on top of this and deposits it there. Now depositing this galactose onto the H antigen makes this person's blood to be blood group B. So this is it, formation of blood group B. Formation of blood group B. Then we got formation of blood group AB. Of blood group AB. Now formation of blood group AB. This one, remember we have our blood group, our red blood cell. Then it has its nucleus. Now, it has already the H antigen. It has already been formed because this was the oligosaccharide material. Then they deposited the fucus. Now the fucus and the oligosaccharide is now our H antigen. Now this chromosome 9 here, in this case, from one parent we got a gene for A. In another pattern from another parent we got the gene for blood group B. So that's why this one will this activity of the gene forming the N-acetyl galactosamine transferase will take place in this one, which came from the A person. Then B, the one which came from the blood group B parent, will form the galactose transferase. So this one will deposit on top here the N-acetyl galactosamine to make antigen A. This one will come and deposit on this second one the galactose sugar to make sure. So this another antigen will be A, another antigen will be B, another antigen will be A, another antigen will be B, like that, million times. So this is how blood group AB is formed. And remember, this one is dominant. Blood group A antigen is dominant. B antigen is also dominant. So everyone wants to show off. So they end up both showing off on the surface of the red blood cells. So this is how AB is being formed. Unlike in blood group, as we saw in blood group A, before we rub off the BB, is that in blood group A, you find that this one from one parent and this one from one parent. So there are two types of blood group A. The genetic information can either be AA or AO. So if this person was AA, it means this one had A gene and this side also came with the A gene. So this A and A, they all made one thing, which is N-acetyl galactosamine transferase, which transferred N-acetyl galactosamine on top of the H substance to make blood group A. But in other cases, you find that this is the first case we have talked about. Now, let us talk about AO. Now, if somebody got A from one parent and another parent gave us O, you find that A is dominant and O is recessive. So this one will not function. O is not function. So it is only A which will produce the N-acetyl galactosamine to form A. So there will be not both of them will make A, but this one will not form anything. This O here, it is recessive and not function. So it will not do any other activity. So the parent will remain blood group A. And the same story is with blood group B, in that the patient, because we saw another one, BB. You can either be BB or BO. Now for BB means that both parents brought B. This one brought B, and this one also brought B. So all of them, they were making galactose transferase to transfer the galactose on top of the H antigen to form blood group B. But in another case of BO, it means one parent brought B, another parent brought O. Now, in case they brought B and O, B will be functional. B is actively functional. It is the one to form B and B enzyme. Because another word, if you are, it is hard for N-acetyl galactosamine. You can easily say this is A enzyme, then this one is, this one B forms the B enzyme, which transfers the galactose to the H substance to form blood group B. Now, this O is recessive, as we said earlier when we were studying about blood group A. So, O is recessive. O will not do any activity. As this one is forming galactose transferase, this one is not forming anything. So, nothing will appear there. Now, in this case, A came from another parent, B came from another parent. Now, in this case here, you find that A is a dominant gene, B is a dominant gene. So, in this case, we say they are codominant genes. So, these codominant genes, they all want to show off phenotypically. So, the genetic material will be A, B, and also phenotypically will be A, B. Unlike this, in this case, whereby the genetic material can either be B, B, or B, O. This one, in this case, also can either be A, or A, O. This case. So there are two genetic ways the A blood group people can present. Even this one, there are two ways the genetic material of their blood group can present. So AB present only in one way, either phenotypically or genetically. So when we look at the formation, of O blood group, O blood group. Formation of O blood group, you find that this is our blood group. I mean, this is our red blood cell with its nucleus. So it has already formed the H antigen on its surface. This is our H substance all over its surface. So the H substance is here, the combination of the fucose and the oligosaccharide. We get the H substance. Now, the H substance is already here on the surface of the red blood cell. Now, if we want to, now it wants to get its blood group. How does it get its blood group? It comes to the chromosome number 9. So the gene which is there on chromosome number 9, it is the one to come and ignite the formation of an enzyme which will transfer certain material and deposit it on top of the H substance. But this O, they are O, because this person would want to be blood group O in case there was any antigen which is either A or B. But being OO, They are all recessive genes and inactive genes. They are not active, and even what they produce, even what their genes produce, is weak, does not do an activity. So they produce nothing, they produce zero. That's why this person is called blood group zero. So initially, these people were being called blood group zero, but some time after, people tried to modify it and started calling them O. But this is blood group zero just because they have zero antigens on the surface of their blood group. I mean, on the surface of their red blood cells. So when you check the red blood cell of blood group A, the red blood cell has A antigen. When you check the surface of the blood group B, the blood group B has antigen B. That's why it's called B. When you check the surface of blood group AB, it has antigen A and antigen B. That's why it's called blood group AB. So when you check the surface of the red blood cell of blood group O person, you find that there are no antigens. There is zero antigen on the surface of the red blood cells. That's why they are called blood group zero. Zero antigen. So they are zero antigen. That's why they are blood group zero. And they modified that by stopping calling them zero antigens, zero blood groups. They call them blood group O people. So if you have any question, we can answer it in a short time. Then we see whether we can have a break. Then come back and continue with more, because we want to tackle the rhesus blood group. So if you are okay with this, we can have some break. Then we continue with the rhesus blood grouping system. Any question? Okay, thank you. And if you want to get more explanation, is in case you forgot to know what, just go to your YouTube channel. I'm going to put this video. So just go to your YouTube channel and look for and type in Dr. PCM Medical Lessons. Dr. PCM Medical Lessons. But when you go there, don't forget to subscribe. You subscribe such that we continue with the study. Thank you. We can have a break.